everyone, I'm happy to be here today to talk about our recent work, Oarfish, which improved the accuracy in long-read RNA-seq transcriptome quantification. First, I want to talk about why long-read and not short-read. So each of these sequencing technologies have their own strengths and limitations. For example, the short-read has higher base level accuracy, higher reproducibility, and high throughput. But on the other hand, the short reads are around 350 base pairs, which much shorter than the average human mRNA transcripts, which are around 2 kilobase pairs. This length discrepancy can cause the fragment ambiguity, which means we will not be sure which transcript this read originates from, and cause the inferential uncertainty. Numerous tools have been developed, such as SAM and Callisto and others, to improve both accuracy and efficiency, but none of these advancements can fully overcome the inherent limitations of the short-read sequencing. On the other hand, we will have the long read, which capture the full length of the transcripts and therefore reduce the fragment ambiguity, but it also has its own limitations, like higher base level error rate compared to the short read and lower throughput. What motivates us here to work on the long read quantification is that in the ideal case, a long read can span the whole length of the transcripts, and therefore the quantification is being directly calculated as the fraction of the reads that uniquely map to the transcripts. But in the reality, this will not happen because of the limitation that long read has. First, we can have the reads that span the whole length of the transcripts, but may not match the full length, which means it may not match the whole nucleotide on the transcripts. And the second is that most reads are shorter than the isoform lengths due to unexpected error breakage during sequencing and other reasons. For example, here in this figure, we show an example of the distribution of the lengths of the reads, isoforms, and also the aligned segment of these reads on the isoform. As can be seen here, the green one shows that the aligned segment of the reads length almost 1.5 times shorter than the isoform length. And also we had a non-trivial multi-mapping rate around 70 to 80%. And we conclude that maybe the probabilistic model used for a short read may also be helpful for the long read. But the problem is the protocol differences between the long read and short read. For example, in the short read, we first do the segmentation on the molecule and then do the fragmentation on those segmentation. But in the long read, we do the fragmentation on the molecule itself. So this fragmentation step on the short read can cause the direct length dependence for the quantification. What we propose here is to remove the length dependence effect in the model and replace it with a coverage uniformity, which will be discussed in more detail later in the presentation. The tools for the long read can be categorized into identification and quantification. Tools like Talent, FLAIR, and Mendellorian are primarily used for the identification. The tools used for the quantification are based on the expectation maximization algorithm, like NanoCount, Bamboo, TransSigner, Isoquant, and Espresso, which each of them use a different conditional probability in their EM algorithm. Here we introduce Oarfish, which will use Lyman score and the read coverage pattern of the transcripts in its conditional probability. Why the coverage model is important. Here we show a toy example that demonstrating how removing length effect from the probabilistic model can increase fragment assignment ambiguity where the human intuition may be clear. In this example, all the reads are equally aligned to both transcript 1 and transcripts 2, but it's clear based on the totality of the coverage, these reads most likely come from transcript 2 than transcript 1, because the likelihood of having this many reads sequenced from transcripts 1, but not sequencing beyond exon 2 is quite low. So we use this coverage pattern model to encode this intuition in our model. In the Lee et al. Paper, they introduced this graphical model that is used for short-read RNA-seq datasets. In this graphical model, the variable read depends on the start position of the read on the transcripts, the transcript itself, and also the orientation of the alignment. And therefore, they obtain the likelihood function based on this graphical model. We modified this graphical model to be applicable to the long read such that the read will depend on the alignment score, the transcript itself, and also the specific start and end position of the read on the transcripts, which this start and end position also depends on the coverage distribution pattern of the transcripts. The likelihood function becomes like this, which has 4 different components. The first component is that the probability of choosing transcript J for sequencing is equal to its abundance in the sample. The second component is probability of having a specific alignment score given that is aligned to transcript J. For this probability, we consider all the alignments of the read, consider the maximum value, have the highest probability, and then the probability for all other alignments will be exponentially decreased as the alignment score distance from the maximum value. And the third component is the probability of start and end position of the read on the transcripts. For this probability, first we have to know how we obtain this coverage distribution model for each transcripts. Here is an example of a coverage pattern of 3 transcripts from alignment of a data set. And as can be seen here, the middle transcripts have a more uniform coverage, which correlates to accurate alignment. So what we are trying here to do is promote the uniformity through the conditional probability of read assignments, such that we push the reads to the regions that have low coverage to make them closer to the average, and pull away the reads from the high coverage region to make them closer to the average. So the key idea here is based on the alignments themselves that also provide some sort of hard constraint that where we can put reads, and we try to have as uniform coverage distribution as possible. And the other approaches, like the 2 papers cited here, also use the coverage uniformity and prove that it was effective in their model, but none of them in the long read context. One function that we can use to obtain this kind of distribution based on the coverage of the transcript is the logistic function. And the procedure that we go through to obtain the distribution model is as follows. First, we align the reads to the transcriptome, segmenting the transcript lengths into disjoint intervals, and then count the reads in each interval. Obtaining these counts for each bin, we can obtain the coverage probability for each segment, such that first we obtain the expected coverage along the transcript lengths, and then use these values to define the input of the logistic function such that it will have a negative value for the highly covered region, and it has a positive value for lowly covered region. And therefore, we obtain the probability for each segment. Now, by having this coverage distribution pattern for each transcripts, we can have the probability of start and end position of read N on transcript J, such that it will be the expected value of the coverage probability across the relevant segments on the transcripts. We evaluate our method on both ONT and PacBio data sets obtained from the NanoSim and TickSim simulators, respectively. To have a more realistic data set, we in these simulations have abundance and error profiles seeded from the experimental data sets in each technology. Here we show the experiment correlation and normalized root mean squared error as 2 metrics for Oarfish with coverage model and without coverage model. And as can be seen here, the coverage model will help the correlation and also the error metrics. And also the Oarfish with and without coverage model have the highest correlation and lowest errors compared to the other methods shown here. We also show the density plot for all of these methods on the only direct RNA sequencing data sets, along with the best fit regression line and Pearson correlation coefficient shown in each of these plots. The x-axis will be the ground truth counts, and the y-axis is the estimated counts by each method. And as can be seen here again, the Oarfish with and without coverage model has the highest correlation and tightest density around the diagonal compared to the other methods. And we also try to do that for the other data sets we had, and we see the same pattern for all of these data sets. We also check the time, elapsed time and memory usage for Oarfish and other methods here, except espresso, because we will not be able to disable the identification module in espresso, unlike that we have that in bamboo. So here, the LR Callisto, which relies on the pseudo alignment instead of the full nucleotide level alignment, has the least time and memory usage compared to the other methods. And in the other methods that are based on the full nucleotide level alignments, Oarfish, specifically in the read-based mode, has the lowest time and memory usage compared to the other methods, which is also comparable to the LR Callisto results. We also evaluate the Oarfish on different experimental data sets from SCT116 from the Singapore Nanopore Expression Project, the dataset obtained from the Tequila-Sick paper, and the UHRR from the PacBio dataset. We only show here the results for HCT116 and UHRR as the representative for ONT and PacBio technology. Unlike the simulated dataset, we do not have the ground truth for experimental dataset, but we can use the short read to compare the long read quantification results. The problem here is that short read quantification is not uniformly reliable across all transcripts in the transcriptome. So here we use the inferential relative variance, which was defined in the Zhu et al. Paper as a metric to stratify the transcripts based on the reliability of the short read quantifications. In practice, the highest INFRV means the highest uncertainty in the quantification, and the lowest INFRV means the most reliable and stable abundance estimate. Here we show the results for only the direct RNA sequencing data sets in terms of the Spearman correlation, Pearson correlation, and also MARD. And each of these categories shown in each of these plots shows the categories of transcripts with different INFRV or reliability. The left-hand side category shows the most reliable transcripts, and the uncertainty will increase as we go from the left to right. The blue line and the orange line are showing the Oarfish with and without coverage model, and as can be seen here, Oarfish has the highest correlation and lowest errors, specifically for the transcripts with highest reliability of abundance estimate. We also do the same for all the data sets, and we see the same results for both ONT and PacBio data set. In terms of the time and memory usage of the experimental data set, again, we see the same pattern as we saw for the simulated data set, except that here for the memory usage, Oarfish is almost the same as LR Callisto. So all of these algorithmic advancements are already implemented in our tool Oarfish, which can be installed via Bioconda, and it can also accept the input file as either FASTQ file or BAM file, and it can track the provenance information of reference transcriptome and compute the inferential replicates for uncertainty quantification. One thing that we want to improve in the Oarfish is that right now we use the static coverage model, which use the fixed coverage based on the initial alignments. This is the limitation that caused the inability to adapt in complex genomic regions for high multi-mapping areas. And therefore, we try to obtain the dynamic coverage model that already used in the short-read tools like RSM, Salmon, and Cufflinks, and proved to be effective. Thanks for your attention, and I'm happy to take any questions.
Any questions? Please move to the microphone.
Nice talk. So my question is, will your method be affected if their annotation is not complete? If there are some novel isoforms that are not annotated in the annotation, so it will usually affect EM algorithm if you cannot identify the novel isoform, right?
What do you mean by not annotated transcripts here?
So my understanding is that your quantification is based on the annotated isoform, right?
Yes.
But if so, in the real data, we know that there are a lot of actually novel isoforms that are not annotated. If you just apply your method to that type of data, will it be affected? Its performance will be affected or not?
So actually, the Oarfish does not do the identification here, but it has the ability to have that you give it both annotated and novel transcripts, and then it can do the quantification on both of those files that you give that to them.
You mean we just added the novel isoform into the annotation, and next we run your method, right?
The novel transcripts cannot identified by Oarfish, but it can be quantified. If
you mean you other method to first identify them, and next you add them into the annotation, right? Next you use this annotation to do the quantification. Is that correct?
Sorry, I don't get.
Maybe you can take those offline.
Okay. Okay. Yeah. That's what I mean. Yeah.
Okay. Another question?
Hi, thank you for the talk. I see that you have estimations of the start and the end. Do you treat in the Oarfish for long read data that there's equal probability to have truncated ends on the 5' on the 3' end?
Sorry, can you repeat your question?
So when you also actually, in one of the shortcomings for long reads, you actually pointed out that the reads, due to the limitations of how the reads are, the cDNA is generated, or in direct RNAs as well, is that the 5' and the ends of the molecules don't always reflect what the true transcript could have been.
Right.
When you model for that, do you model equal probability that those degradations will happen both on the 5' and the 3' end?
Actually, the simulated data sets that we have doesn't consider that degradation on the 3', 5' end. So we already prepare another paper that works specifically on the
Thank you.
3', 5'.
Thank you so much. Let's thank the speaker again.
Thank you.